So actually, many of the section of my presentation has been covered by Ali as well as also Audrey earlier. So I'll just jump directly. So this is an outline of my presentation. So I would cover insecticide classification, mode of action, resistance, and also resistant management, and lastly on Probit analysis. So the world population is going to reach 8 billion people by the year 2025, and in order to feed and clothe and also to prevent vector-borne diseases, so food crop yields need to be increased, more natural fibers are required, as well as also reducing the insect vector population. And all these would not be able to achieve without the use of pesticide. So in the human environment, there is a dilemma. So we do not want to have any pests in our environment, while at the same time, we also do not want to have any pesticide. So if you look at the classification of synthetic insecticide, they are actually numerous. If you look at the IRAC classification, they are actually more than 30 different mode of action. But what I'm going to do is I'm going to cover those that are highlighted in blue in this presentation. So mainly the chlorinated hydrocarbon, the OPs, the carbamate, the Pyrethroids, Neonicotinoids, the insect growth regulators. So one of the I would say common group of insecticide used against vectors that is popular in the past is actually chlorinated hydrocarbon, and started with the discovery of DDT back in 1939, and they are usually chemical unreactive and not water soluble, and that makes them very persistent in the environment. And most of the candidate actually has been banned for usage, with exception in some countries. So, and the reason is because, especially for DDT, is because of its stability and also fat solubility, and sublethal doses could be ingested by animals and will not be metabolized, but instead would be stored in body fat. And because of this persistency, it can actually move up through the trophic level to the top of the trophic level in a phenomenon known as biomagnification. The second group is organophosphates, and this was first discovered by the Germans during World War II as a toxic agent in the form of nerve gas for chemical warfare. And subsequently, after the war, more and more compounds are being discovered and later being used as an insecticide. Now, OPs are more toxic to higher animals than chlorinated hydrocarbon. They are also chemically unstable and non-persistent in the environment. So you can identify OP with the presence of a phosphate group, and the candidates that have been widely used, including the chlorovos, malathion, as many are familiar with, chlorpyrifos, diazinon, etc. So majority of the OPs are actually contact and also oral poison. So this is one incident that happened in Tokyo back in '95, where a cult group actually launched a sarin gas attack, and there are quite a number of people that were killed, and more than 1,000 people collapsed in the Tokyo subways. So if you look at the different types of organophosphate, essentially you can divide them into 3 subgroups: the aliphatics, the cyclic, and also the bicyclic. Essentially, the aliphatic ones are the ones that is lacking the ring, where the cyclic one has 1 ring compared with the bicyclic with 2 rings. So, for example, malathion, it's the aliphatic within the aliphatic type, and the chlorpyrifos, diazinon, and phenyltrethion is in the cyclic group. So the interesting part with OP is they will be bioactivated by cytochrome P450 to form the oxon group. So as you notice here, unfortunately, I'm not able to use my my pointer. So what happened is, if you look at the copariphos, basically when it formed the copariphos oxon, it is actually more toxic. And malathion, when it is activated to form malathion oxon, it is actually a more toxic form. So next is actually the carbamate, and carbamate was first discovered in 1951, but subsequently, at that point of time, it was not effective, and because of high costs, not many are using it. But subsequently, with the discovery of carbaryl, they become increasingly popular. And today you have bendiolcarb that is still being used in vector control. So carbamate is identified by the presence of carbamic acid ester, and they have high mammalian toxicity and usually broad spectrum for insect control. So if you look at the types of carbamate, it's very similar to OP. So you have the aliphatic group and also the cyclic group. So the cyclic group would have a ring within the structure, compared with the aliphatic group that do not have a ring. Next is actually pyrethroids. So pyrethrum, which is a botanical insecticide, was extracted from chrysanthemum flower. That shows good knockdown back in the 40s, but it is too costly to extract, and of course unstable under the sunlight. So based on the chemical structure of the component within the pyrethrums, so you have pyrethrin, you have sinitrin. So the chemical analogs of pyrethrum will actually synthesize. Now, as you notice the name Pyritroids. So whenever you see the word oid at the end, it is actually means it look alike. So I'm sure that many are using Android phones besides iPhone. So when there is an OID, it means it's alike. So Pyretroid means pyrethrum alike. So if you compare between pyrethrins and pyrethroids, you can actually see that you can actually identify based on the acid moiety that contain 3 carbon ring in a triangular shape. So there are a total of 5 generations of pyrethroid. The first generation was available commercially back in 49. This is elytrin. It was elytrin. It's effective only against flying insects such as flies and mosquito, but not against cockroaches. They are photo unstable as well. Second generation, they have better knockdown against flying insects. You can also synergize it with other compounds. The representative candidates such as tetrametrin was introduced in 1965. You have subsequently resmetrin, bioresmetrin, etc. Again, second generation and first generation, they are both photo unstable, so they will degrade readily under UV light. So the third generation started off with the introduction of permethrin and subsequently familaric. So finally this group, they are photostable and they have good residual property, so they could last for a while. They also have exceptional killing activity or insecticidal activity. The fourth generation is actually 10 times more active than the third generation, and they are photostable. They have good residual and superb insecticide activity. And you have candidates such as deltametrin, cypermetrin, cyphotrin, etc. Okay. And finally, the fifth generation, it's a volatile pyrethroid. So you have candidates such as metofultrin, and they could be used as it is without even the need, and widely used, especially in household insecticide. So if you look at the types of pyrethroid, you can divide them into 3 types. The first one, it's without the alpha cyano group. And the second one is with the alpha cyano group, as I highlighted in pink over in my slide. I'm sorry, I can't use the pointer. But with the alpha cyano group, they have better killing property, okay, and also they are stable under UV light. So the last type, that is the volatile group, they have this fluorilated benzene ring, as you can see, methylfluorine and also transfluorine. Now, next group is actually neonicotinoids. As the name says, it resembles natural product, nicotine. So you have candidates such as imidacloprid, acetamiprid, dinotefuran, and clothianidin. These are the candidates within neonics, and they have low mammalian toxicity. And if you look at neonics, oops, what happened? I can't advance. Okay, so there is a structure of or nicotine-like structure within the compound of neonicotinoids. Next, it's the insect growth regulator. So these are chemical substances that will disrupt the insect growth and also the development, resulting in the death of the insects. Essentially, there are two types of IGR. The first one, known as juvenile, or sometimes we call it JHA, juvenile hormone analogs. And the second group, it's actually the chitin synthesis inhibitors, and also known as benzophenolurea. Okay, so juveniles, these are structurally similar or resemble the insect juvenile hormone. So you have candidates that are used in mosquito control, such as metoprene and also pyriproxyfen. And for the chitin synthesis inhibitor, this would inhibit the formation of chitin, which is an important component of insect exoskeleton. So I'm sure that many of you have heard before of dimilin, diflubenzuron, okay, which is used for mosquito control, and a few other types of CSIs. So this is the first slide actually show Culex affected by metoprene. So the larvae died when the adult is trying to emerge from the pupa. And the second slides below show the cockroach affected by phenoxycarp. So as I mentioned before, they are natural juvenile hormone alike. So many of these candidates are widely used in vector control as well as also urban pest control. Next group, it's the microbial insecticide. So we have the most common one used in vector control, Bacillus thuringiensis. They are actually a number of different strains of BT, and usually commercial BT products are powdered mixture of dried spore and also the crystalline delta endotoxin. So both the spore and also the toxin crystal are produced within the vegetative cell of the bacteria. And if you look at these different strains of BT, the one that is commonly used for mosquito control, black fly control, it's actually BTI, okay, Bacillus thuringiensis israelensis. So they are also, of course, also Bacillus sphaericus being used against mosquito, especially those mosquito such as Culex, Mansonia that breed in polluted water. Okay, spinosad, on the other hand, is under the group of spinosyn. This is an insecticide mixture of spinosyn A and D, and this is basically derived from soy actinomycetes under the aerobic fermentation condition. So let us move on to insect neurobiology. I'm going to go slower with this one. Now, so if you look at the insect nervous system, it basically composed of a series of interconnected cells. These are known as neuron, of which the electrical charges, known as an impulse, would travel across the neuron. So impulses, of course, are carried from one end of the nerve cell across through the synapse to the next one by a chemical messenger known as neurotransmitter. So if you look at a neuron, it consists of a cell body. Within the cell body, you have nucleus, you have the mitochondria, and then you have the axon, and the axon will end with synapse, and the synapse would then get so-called very close to the other synapse over the next axon, okay? And for the impulse to travel through the synapse from the pre to the post synapse, you would require the service of the neurotransmitter. So the signal, of course, would enter through the stimulus into the nerve cell and travel through the dendrite, and they travel electrically along the axon. And then when they reach the junction, then the neurotransmitter will be released, and then the neurotransmitter will travel across to the other end, bind to the receptor, and subsequently produce another impulse and travel and so on. So if you look at the axon itself, okay, just look at the axon first. So what happened is under resting potential, you have more sodium on the outer of the cell and more potassium in the inside. So when a stimulus is being given, what happened is the sodium gate would open and the sodium would move in, and that would actually create an action potential. And subsequently, you have then the potassium gate open, and the potassium would travel out while the sodium channel gate would close, and then they will be back to the resting potential. On the other hand, when the impulse arrives to the presynaptic cleft, what happens is it will activate the vesicle to release acetylcholine. And as you can see right here over the two, is shown there over two. So what happened is the acetylcholine would be released, and acetylcholine would then travel through to the other side and bind to 3. That is the acetylcholine receptor. And upon binding, it will trigger an impulse. And after the job is done, then you have the acetylcholine esterase would remove acetylcholine or degrade acetylcholine from the receptor, and subsequently it will get absorbed back into the cell in the form of acetate and choline. So this process, of course, happened very quickly, and you don't even feel anything, okay, that such things are happening. So in terms of neurotransmitter, it's not just acetylcholine, but they are also GABA, okay, that is gamma aminobutyric acid, and also glutamate. But for the purpose of this lecture, I'm only going to talk about acetylcholine. When we discuss in future about fipronil, probably we will talk about GABA as well as also insecticides such as ivermectin. We'll talk about glutamate. But now neurotransmitter are released from the presynapse region, then move across the synapse, receive over the postsynapse region at the receptor site. And when the job is done, then it will be removed. So let us look at how different insecticide kill. So if we were to divide insecticide, we can generally divide them into 2 main groups. The first one are the one that target insect nervous system, and the second one are the one that do not target insect nervous system. So within the one that target the nervous system, we're going to discuss just the one that target the sodium channel and also the one that target the acetylcholine system. For the one that do not target the insect nervous system, we're going to discuss about insect growth regulator or IGR, and also the elementary toxin. So let us take a look at the first one. So the first target is sodium channel, as has been mentioned by Ali and also Audrey. So you have chemical class such as DDT, Pyrethroid, pyretrins. These are mainly the one that attack the sodium channel. So both pyretrins and Pyrethroid disrupt the normal nerve function in axon. So remember earlier that I showed the slide where the sodium channel are open and allow the sodium to move in. So once the job is done, then the gate would close and then there will be no more sodium coming in. But what happened with DDT and Pyrethroids, they will hold on to the gate and the gate will remain to open and allowing more and more sodium to flow in. So this would cause uncontrolled and uninterrupted nerve firing, and that will result in tremor and of course shaking in the insects. So if you look at this particular slide, okay, under normal situation, you have the sodium channel open, and once the job is done, then the gate will close. But once you have the insecticide, imagine that the insecticide is holding the gate and prevent the gate from closing. So the sodium ions would keep flowing in, and that will continue to trigger impulses. The next target site is actually the acetylcholine system, and this would be referred to insecticides such as OPs and Carbamate. And what happened is, as I mentioned before, once the acetylcholine move over to the receptor and the job is done, then the acetylcholine esterase will then remove the acetylcholine from the receptor, okay, to prevent it from continue to trigger impulses. But what happened with OP and Carbamate is they will inhibit the function of acetylcholine esterase enzyme from doing their job, okay? So because acetylcholine esterase is no longer functional, acetylcholine would continue to bind to the receptor, and that will continue to produce impulses and causing the insect to eventually die of hyperactivity and also paralysis. Next group is actually the neonicotinoid. They also attack the acetylcholine system, but neonicotinoid is not attacking acetylcholine esterase. Instead, neonics is mimicking acetylcholine. Okay, so acetylcholine is a neurotransmitter that will bind to the acetylcholine receptor, and neonicotinoid, they are also capable of binding to the acetylcholine receptor. And because they have been binding to acetylcholine receptor, acetylcholine will not be able to remove neonic from the acetylcholine receptor, and causing continual impulses would be produced, and this would be causing tremor and hyperactivity. As you can see with this German cockroach here, that is actually not a very pleasant way to die. Okay, so they will continue when they get intoxicated, they will continue this movement all the way until the insect die. Next is actually the insecticide that do not target the insect nervous system. So we talk about the first one, insect growth regulator. So juvenile hormone analog, as we all know, as the insect gone through the immature stage all the way until adult, so the level or the title of juvenile hormone would drop. Okay, and this is more pronounced, especially in insects that undergo complete metamorphosis, such as mosquito. So what happened is, as at the stages of larvae, they have higher level of juvenile hormone, but once they move on to pupae, the level of juvenile hormone will gradually drop. And as from pupae to become adult, the level of juvenile hormone would drop even more. So But when you provide them with juvenile hormone, this would keep the immature stage from becoming adult. Okay, and what happens is when the level is such that it does not kill them right away, as they try to emerge to become adult, they will die at that stage. Okay. On the other hand, you have chitin synthesis inhibitor. So this would block the function of chitin synthetase. This is an enzyme that is responsible to convert some of the chemical compound into chitin, and chitin is actually an important component of insect exoskeleton. So if you look back at this particular slide, so for insects that actually go through many stages of molting, chitin synthesis inhibitor is probably a more suitable compound, okay, such as cockroaches or termites. They have to go through many different molts. These are insects normally that undergo incomplete metamorphosis. But chitin synthesis inhibitor, of course, could also be used against mosquito. Next is actually the insecticide that act as alimentary toxin. And for this one, we talk about BTI and BTI H14 spores, and also the Paraspora crystal when you actually do larviciding. So it will first be ingested by the mosquito larvae. And following ingestion, what happens is when they get into the mosquito gut, larvae gut, so the meat gut actually has a pH of 9 to 10.5, and that would trigger the endotoxin to be released. Okay, so when the toxin is being released, they would then bind to the insect gut cell, and this would form a large pore that will eventually increase the water permeability into the cell membrane, and subsequently causing large uptake of the water. And if you look under microscope, what happened to the larvae? Subsequently, it will rupture the gut cell, and you would see the larvae get killed. So for BTI, human gut pH is acidic. Okay, normally below 6, and because of that, theoretically, when you drink BT, okay, the impact would not be there, unlike to a gut with an alkaline gut pH.
Professor Lee, your first leg of the talk is going to be up in a couple of minutes. So, and then we have only a 10 minutes break, and then we join you again for your talk. Is that all right?
Sure. Yeah. Yeah.
Thank you very much. Thank you.
So Audrey has talked about synergists. So synergists is actually a chemical that do not have any inherent insecticide activity, but it could actually inhibit some of the function of the enzymes. So when some of the function of the enzymes get inhibited, this would enhance and also increase the effectiveness of the insecticide. So synergists actually could be used to block some metabolic processes. Okay, and you would find that on this slide, there is a number of different synergists. For example, piperonyl butoxide, PBO, that is used to inhibit the function of cytochrome P450. We also, in insecticide resistance studies, sometimes we also use DEF, and DEF is actually a fungicide, and that could actually inhibit the function of esterases. There have also been attempts to use DEM as a synergist to look at GST activity. So with that, I'll probably end my first half of my presentation. So I'll continue the next with insecticide resistance and also Probit analysis. Thank you.
Thank you very much, Professor Lee, for this very informative and nicely laid out presentation. It actually took us some 30 years back when we had the lectures of toxicology and related courses at the university, and I hope everyone of our participants enjoyed as much as I did. Okay, then, so everyone, we have 10 minutes for a quick break. Please don't go far away. We start at 10 minutes sharp for the second leg of Professor Lee's lecture. Thank you very much, everyone. Okay, I think the 10 minutes is up. Professor Lee, please, can you start the second part of your presentation? I hope everyone is there. Okay, the floor is yours for the next 30 minutes.
Thank you, Ali. All right, so welcome back. So let us talk about insecticide resistance. So insecticide resistance, if you look at the definition by WHO, this is an ability of insects to survive exposure to a standard dose of insecticide. So of course, this is because of physiological and behavioral adaptation. But if you look from the operational definition by IRAC, this is a heritable change in the sensitivity of a pest population that is being reflected by repeated failure of a product to achieve the expected level of control when you use it at the label recommendation for that particular pest species. So there are two definitions. So one is more, I would say, from the sample of the insect that you collected and you test it using a standard dose, while the second one is more operational. So there are, of course, a number of very important publication, and Audrey has mentioned this before. And the one that was published in PLOS Neglected Tropical Disease by Moyes, that is one of the important paper that you should try to get hold of and read if you have not done so. So insecticide resistance is actually happen because of selection pressure. So under a wild population of insects, they are bound to have some individuals within the population that are carrying the resistant gene. And as you continue the same insecticide over and over, so you will select out those resistant individuals. And of course, you would kill a substantial number of susceptible individuals within that population. But as they continue to mate and multiply, you would find more resistant individuals within that population. And if you continue to select it over many different generations and up to a point, you would find that predominantly the population consists of insects that carrying the resistant gene, and that would give you a control failure. So in terms of mechanism of resistance, this again has been mentioned before by the previous two speakers. So I will not go very detailed in again. So generally, there are 4 different types of mechanisms. So the first one is behavioral resistance. Then you have penetration resistance, metabolic, and lastly, target site resistance. So the first two, behavioral resistance and also penetration resistance, they are considered at this present moment as minor resistant mechanism, whereas the last two are considered the major resistant mechanism. So if you look at where they target, so behavioral, it's of course refers to the behavior of the targeted pests. And for penetration resistance, it is actually at the insect cuticle. The metabolic resistance is referring to the detoxifying enzymes. And lastly, the target site is the site where the insecticide are acting. So behavioral resistance, generally, these are modification in insect behavior to avoid the lethal effects of insecticide. There are 2 types of responses. what we call irritancy, where insects will leave the insecticide treated area, could be after making physical contact with the chemical very shortly or even without. And then you have what we call repellency, where the insect move away from insecticide treated area without even making any contacts. So next, it's actually penetration resistance. As Audrey mentioned before, this is referring to modification of insect cuticle that become thickened. So it will slow down the absorption or the penetration of insecticide into the insects. This is, again, at this moment of time, consider a minor resistant mechanism. So what is the major one? So metabolic resistance is a major one. These are changes in enzyme system with the increase of enzymes or shortened detoxification process that will result in rapid detoxification of the insecticide compared with a normal situation. So this would prevent the insecticide from arriving to its targeted site or reducing the amount of insecticide from arriving to its targeted site. There are three major enzymes involved. You have the oxidases, mainly the cytochrome P450. You have the esterases, and lastly, GST or the glutathione S transferase. So when it comes to metabolic resistance, so we have, for example, two major groups of insecticide, Pyrethroids and organochlorines, mainly DDT, and you have the OPs and Carbamate, another group. Okay, so the detoxification enzyme would play its role to detoxify this insecticide once it gets into the insects. On the other hand, you have target site resistance, and these are mainly due to genetic mutation that altered the target site, so the insecticide no longer could attack or bind to the targeted site. As we have learned before this, so you have Pyrethroids and DDT acting at the sodium channel. You have acetylcholine esterase, where it's being attacked by the OPs and also Carbamate. So target sites resistance normally refers to some form of mutation at a certain point where it will substitute the amino acids with a different amino acids. Okay, so I give you an example. For example, if you look at the slide S989P, okay, S989P, so S refers to serine and P refers to proline. So what happened is at this particular point of 989, the amino acid serine has been replaced by proline. So how does this happen? Okay, so if you look at the nucleotides that will coat the amino acids, so if you look at the highlighted one, so proline, you have CCT. So when you have just one nucleotide change from CCT to TCT, it will change the coding. Sorry, serine TCT, when you see TCT and when you have one nucleotide change where the T is replaced by C to CCT, then from serine, it will encode proline. And this is actually what happened to that point mutation. So for target site resistance, as mentioned before, there are two types of target site resistance in mosquito. There are more in other types of insects, but for the purpose of this course, we're only going to discuss these two. So you have the modified sodium channel, where it has reduced sensitivity to Pyrethroids and DDT. And then you have also the altered acetylcholine esterase, where it will have an impact on OPs and also Carbamate. So KDR resistance, this of course confer reduced sensitivity to Pyrethroids and DDT. And this is actually basically an allele or a particular site where it has been changed or mutated, and that basically causes KDR resistance. But having said that, when you screen through different population of mosquito, you may find some of these so-called points that has been changed, but it does not directly imply that it will confer KDR resistance. So sometimes KDR resistance could be due to one mutation point or multiple mutation points. So if we look at the role of different enzymes and targeted sites modification to resistance, so we have these 4 groups of insecticide: Pyrethroids, OP, Carbamate, and also DDT. So each one of these resistance could be conferred by either metabolic or target sites. And again, this actually has been shown before by the previous 2 speakers. The other aspect that is important to know is cross-resistance. So between different classes of insecticide, because they may share the same mode of action, so you may find that using one classes of insecticide, which of course the insect has become resistant to, when you start switching to another group of insecticide or another class of insecticide, if both of these classes of insecticide are sharing the same mode of action, you may find less effectiveness of this new group of insecticide that you use. And of interest, as shown here, Carbamate and OP, and they are sharing the same mode of action, so that could confer also resistance to the other group. And vice versa with Pyrethroid and DDT, both could confer resistance to one another. But it may not always be the same, because this would only happen when you have target site resistance. But in the case of metabolic resistance, so when you have metabolic resistance, for example, resistance to Pyrethroid, when you start using Carbamate, for example, you may also find resistance to Carbamate, because cytochrome P450, for example, could also confer resistance to Carbamate. So let us take a look at the principle of insecticide testing and Probit analysis. So the interaction between insecticide and also the living organism, they are dose related. Okay, so when they are dose related, it means that, so when you provide an insecticide at an increasing amount, you would find that more and more insects would get killed. Okay, and normally toxicity is expressed in the form of LD50. This is the amount of toxicant that will kill 50% of the test organisms. And of course, you can express them in the form of milligram per kilogram body weight of test animal, especially when you are using rats. If you are using, for example, cockroaches, we normally express them in microgram over gram body weight of the test insects. But in many situations, we are not able to determine the exact dose that the insect contacted. Okay, for example, you are testing mosquito larvae, okay, larvicide in the water. So you're not able to know how much of insecticide that the insect contacted. In that situation, you will then express them in the form of LC50. That is the concentration of the solution that you place your larvae in, okay? And that is the lethal concentration that kills 50% of the test insect. Sometimes you can also express toxicity besides using dose or concentration in a form of time. And that is more so, especially when you are using, for example, WHO test kit. So you have, you know, an impregnated paper, insecticide in impregnated paper. You release the mosquito into the tube, and you assess mortality of the mosquito with time. And from there, of course, you could also determine LT50, besides, of course, mortality after a certain period of time. So that would be the time taken for a certain concentration of insecticide that cause 50% mortality of the test insects. So when expressing with time, normally we will use the one standard concentration, okay, or one single dose, and that will be standardized. So when you have higher LD50, that means that the insecticide toxicity is reduced. Okay, for example, if you find LD50 for cypermethrin is 500 milligram over a kilogram, and LD50 for Timifos is 8600 milligram over a kilogram, that means that cypermethrin is more toxic than Timifos. Okay, so that is the basic principle of insecticide toxicity. Now, what is Probit analysis? Okay, I know for the fact that some of you have lots of experience with Probit analysis. Some of you may not have any experience with Probit analysis. I know for the fact that maybe on Thursday, Dr. Ennis Koo and also Audrey Lenhart would be discussing about insecticide screening and also Probit analysis. So I would not discuss in detail here, but I would give you some basic principle what Probit analysis is all about. So it is actually a statistical procedure that is used in analysis of bioassay in a sequential mortality in a range of doses that result in a sequential mortality increase of the pests. So the insect normally would respond to the insecticide action based on a normal distribution. So Probit analysis is actually an idea that was initiated by Chester Bliss. He's an entomologist in Connecticut Agriculture Experimental Station back in the 30s. So he was intending to test pesticide that control insects that feed on grape leaves. So when he plot the insects to pesticide, he can actually see that the pesticide affected the insect at a different concentration, but the curve, he's actually obtaining a curve that looks like that. So the problem is because it's a curve and it is not a straight line, so it is not possible to determine what is the particular concentration that able to kill, for example, 50% of the test insect. So Bliss actually developed the idea of transforming the sigmoid dose response curve to a straight line. And it was not until the 50s that David Finney, a professor of statistics at University of Edinburgh, then took Bliss' idea and then wrote a book known as Probit analysis that we use today. Okay, so a typical distribution for a tolerance concentration, you would find a curve that looks like this. Okay, so the concentration on the x-axis, if you were to turn it into log 10, okay, it will give you a normal distribution curve. Okay, and subsequently, if you take the mortality of the insects, okay, and have it in percentage, and you do accumulation of mortality of the insect over the y-axis, and of course, if your x-axis is still concentration, you would find a sigmoid curve from here. And if you were to turn the concentration over the x-axis into a log 10 and having the cumulative mortality, percentage mortality over the y-axis, you will find a normal sigmoid curve. Now, this is still a curve, okay? So how do you turn this so-called normal sigmoid curve into a straight line? So that is to take the constant cumulative percentage mortality of the insect over at the y-axis, you transform the percentage into Probit unit. Okay, so in the past, okay, so we would be using a table, okay, to do the transformation. Okay, but these days you can use computer programs, so you don't have to worry too much about it. But why I'm telling you this is because I want you to know the principle behind it. So for example, if you want to transform 11% mortality, so you would find that second row that is 10. And then you see the third column, 1, you bring it down to the second row, that is 3.77. Okay, so that is your Probit unit. And if you look at 50%, okay, 50%, so 50 over the first column, you go down all the way to 50, and you look at the second column where it is 0. So 50% will give you Probit unit of 5. So when you plot that, okay, to your y-axis, you will get Probit unit, and you get log 10 concentration, you will get that straight line. So from that straight line, you can then determine what is 50% mortality, that is the Probit unit of 5, and then you can then determine the concentration that will give you. But this is the old school or old ways of doing it, you know, when you don't have a computer program. But need not worry, these days there are a lot of computer programs available that you could download it from the internet. Some are actually free. Okay, there's a free software in Excel form that I have actually posted the link in chat, okay, earlier before I start the presentation. So you could scroll up and try to go to that website and download that program in Excel if you have no access to any of these so-called Probit analysis program. Okay, so a few notable ones. So you have Polo Plus or Polo PC. This is actually a very common one used by many of the US researchers. You could also have them in statistical package, whether it's in SPSS, in JMP, even the old SAS. And these days you can even run Probit in R. Okay. So how do you interpret toxicity results? Okay. So bear with me for those of you who is familiar with this, but for those of you who are not familiar, let us take a look at the toxicity result. So there are a few columns that you would find in any toxicity result. The first one, of course, over the far left is actually the first column is on insecticide that you are testing. It can be also the strain of insect that you are testing, and refers to the number of insect that you use for the evaluation. So if you use a total of 100 insects, that will be written there as 100. Okay, so 100 refers to the total number of insects that you use in the entire experiment where you generate that particular LC50. Okay, next is, of course, the toxicity result. So you have the LC50 and also LC95. And notice over there, there is a bracket, okay, that gives you a range, okay. So for example, LC50 for A, 1.9 with a 95% fiducial limit of 1.7 to 2.1. And you have LC50 for B, 2.3. You have fiducial limit of 2.0 to 2.6. What's that? What does all this mean? I'll explain in a short while. Okay. Then you have the slope. Okay. Now slope, of course, refer to that Probit line that you generate earlier. So remember this line that we generate earlier. So how steep is this slope would be represented here under slope. And then you have chi-square value. Okay, chi-square value is the goodness of fit. Okay, so goodness of fit refers to how well that fit is. Okay, or rather, should I say, if you have a line that looks like this, you notice that the line did not pass through every single point. Okay, so it may pass through some points, but it bypassed some others. Okay, so the deviation between that line to that point, you will then calculate it in a form of chi-square. And if it deviate too much, for example, all this X point that It's on this graph. If it's too far from that line, so imply that whatever that you observe and whatever that you expected, when it's deviate too far away, that will give you a larger chi-square value. So it means, so the larger the chi-square value, that means that your fit, it's not good. Okay, so you try to get it as tight as possible. So how do you do that? Basically, you also have DF, that is the degree of freedom. That means that how many points that you use minus 2. Okay, for example, in this case, 1, 2, 3, 4, 5. Okay, you have 5 points. So your degree of freedom is n minus 2. So that is 5 points minus 2. Okay, so degree of freedom would be 3. Okay, so in this case, when you have degree of freedom of 5, this implies that you are actually using 7 points to plot that graph. Okay, to plot that line. And you would take chi-square, divide by degree of freedom. That will give you what we call heterogeneity factor. Okay, so in this case, this would be around the range of 0.6 to 0.7. So as long as you are keeping your heterogeneity factor below 2.0, you are basically having quite a good fit. Okay, so next one is actually to look at the toxicity data and trying to make sense whether they are significantly different from one another. Okay, for example, for insecticide A and B, okay, so if you look, so how do you tell whether they are significantly different from one another? You look at the easiest way. Of course, there are many different ways to do it, but the easiest way is to take the fiducial limit and see whether they overlap with one another. Okay, take the LC50 95% fiducial limit. So you have 1.7 to 2.1, and then B 2.0 to 2.6. So in this case, they overlap. Okay, so when they overlap, it means there is no significant difference from one another. Okay, so in the case of C, you have 4.2 to 4.8. So compare A with C. So A is 1.7 to 2.1. C is 4.2 to 4.8. They do not overlap. So it means that there is significantly different from each other. Okay, so this is one way that you could compare between toxicity. Of course, if you are working with insecticide resistance, so you could also be taking LC50 of A and B, for example. If A and B and C represent different strain, for example, A is your susceptible strain and B is your field strain, then you will be taking 2.3 divided by 1.9, and that will give you the resistance ratio. Okay, so that is another way of doing it, but I'm not going to discuss it right here because that is more practical aspect. Now, Abbott's formula.
You have 4 minutes to wrap up your talk, please.
Yeah,
Thank
yeah,
you.
almost there, almost there. Okay.
Thank you.
All right. So sometimes, okay, in your untreated group, you actually see mortality. So what do you do with that? Generally, if the mortality rate is below 10%, that's fine. You can use Abbott's formula to do the correction. So it has to be below 10%, okay? But if it's more than 10%, you then repeat the entire experiment, okay? So for the third point, okay, if the mortality rate is less than 10%, not 20%, okay, it's a typo there. It should be 10%. So you would use this Abbott formula and you would correct the mortality and before you run Probit analysis. But these days, because you're using computer, so all this would be done by your computer, okay? But you have to put in the mortality rate of your control. Now, what are the common pitfalls in testing? Okay, first of all, it's not using standardized test insect. I know if you are doing monitoring in the field, it is very hard to do that. Okay, if you're collecting mosquito, for example, from the field and you want to test them right away, it is not possible to get standardized age. Okay, but if you could do that, if you are going to collect them back to the lab, you're going to rear them into F1, then you have more points and you have more opportunity to standardize your test insects. So if females have been mated, have been blood fed, there could be some discrepancy in your result compared with those that have not blood fed. So strain, of course, play a major role. If you are raising mosquitoes in the lab, you have to standardize all the medium that you use for your food, because different foods that you use will matters. And of course, if you are rearing mosquito in different trays, the tray size matters, and also the rearing density also matters. And lastly, it's the recording. Okay, so you need to have a standardized criteria to score for knockdown. Okay,
أنا عندي زوم ميتنج عقبال عندك.
Because you may have different personnel scoring knockdown of your test insects, and each one of them may use a different criteria, and that could cause some discrepancy in your recording. So with that, that's all. This is my last slide, lab or field tests. So if you're using lab tests, this is cheap, this is fast. You could standardize your test insect. You could also compare with a susceptible strain. But if it's a field situation, when you are testing in the field, this is a real situation. Your insects are mixed between susceptible and also resistant insects. Generally, field tests are more expensive to be done, time consuming too, especially when you are testing an insecticide product. You need to have enough replicates before you can get good results. So with that, this is my last slide. Thank you so much for your kind attention.